Now, this is all fun and games for the virus, but what does it actually do for the host cell? Being infected by a virus is very damaging to host cells. We often see cytopathic effects or CPEs. These is any damage that has been done to the host cell. And often you can see the damage very clearly when you look at it on the microscope. Now, keep in mind, most microscopes will not show you viruses in clear detail, but we can see their indirect effects by seeing cytopathic effects. So you might not be able to see, hey, there's one viral particle there, but you can see, wait a second, that host cell looks a little off. Often an infected cell will undergo changes such as they may be much larger, so they'll be bigger in size. They may have less organized cell shape. They often have inclusion bodies or compartments where they've stored materials, or they may have damaged organelles. Additionally, sometimes they will form a syncytium. So some cytopathic effects will cause host cells to fuse with each other. And what you end up with is this big blobby sort of host cell that has multiple nuclei. And normally we think one cell, one nuclei, but if a singular piece of cytoplasm has multiple nuclei, it's known as a syncytium. Okay, so here are some of the cytopathic effects you might see. Giant cells with multiple nuclei as opposed to the normal cells. So they may be much larger. If you stained for DNA, you'd notice that there's multiple nuclei in one cytoplasm. You might also see inclusion bodies where they've stored viral particles, trying to get rid of them frantically. And if we look over here in the cartoon, you see that there's a regular monolayer on the, if you're growing cells in a dish of host cells, and they look all happy, like with some connections, but not too much. But you can see if they're infected with poliovirus, oh, we completely killed them all. There's nothing on your dish of cells anymore. Or maybe you see that it's infected with an adenovirus and wait, they're much more round, they're clumped together. We can't see their nuclei as easily. Or maybe they have multiple nuclei within one large blob of cytoplasm. Or if we come down to the bottom here, you might notice that there's inclusion bodies, this dark burgundy, along with the regular nuclei. Most damage will kill the host cells, but sometimes viruses can maintain a carrier relationship, one where the host cell isn't permanently damaged by the virus. It isn't immediately lysed, but instead kind of has a persistent low-level infection of harboring the virus. Persistent infections can last several weeks. Some can last for the remainder of the host's life. Viruses can remain in the cytoplasm, so they can just hang out outside the nucleus, or sometimes they incorporate their DNA into the host genome, save it for later, and then they can kind of come out of retirement later. This is known as a provirus. So some examples are herpes. So cold sores, maybe on the lip, you can have one for a little while with an active infection. It'll go away for a little while, but then it might pop back up again. Or chicken pox. Children often get chicken pox and eventually it goes away. And then decades later, as a senior citizen, shingles comes back. The provirus basically comes back. We also see that in addition to infectious diseases being affiliated with viruses, cancers are as well. Some viruses permanently change the genetic material of the host. And as we know, mutations can often lead to cancer. Any sort of changes to the genome are worrisome and therefore something to be avoided. Roughly 13% of cancers are thought to be caused or connected to viral infections. A couple of key terms that often come up when we talk about viruses and cancer are transformation. Transformation is when naked DNA or DNA that's not packaged is transferred from a donor to a recipient. So maybe a host cell sees some DNA from a virus kind of floating by and they bring it in and put it in their genome, and oops, maybe that wasn't a good choice. Oncoviruses are viruses that will initiate tumor formation, viruses that will change gene expression or maybe turn on oncogenes to proliferate, have aberrant growth. Some examples of viruses that are linked to cancer are the papillomavirus or HPV. You may have heard about it in relation to the HPV vaccine. This is a cervical cancer. Hepatitis B is affiliated with liver cancer, or herpes virus has been connected with lymphoma. There are three main mechanisms for viruses causing cancer. Some retroviruses can cause cancer, and they do this by incorporating oncogenes into the host DNA, which leads to overgrowth. So they come in and infect their host, insert their DNA into the host DNA, and then as that gene gets expressed, it makes viral proteins that cause cell overgrowth. You could also have retroviruses affect the expression of host oncogenes. So maybe there's a gene that's related to embryology or something, and we want it, yes, when you're a baby, you're growing a lot, but you turn it off in adulthood. Oh, well, maybe the virus came in here and flipped the switch back on. So suddenly you're making a protein that you shouldn't be at the wrong time. And this can cause overgrowth. You can also have DNA tumor viruses. This one kind of skips a step. Instead of incorporating it into the genome, it just automatically expresses the viral DNA. It makes viral proteins, which lead to cell overgrowth. All of these are methods or mechanisms for viruses causing cancer.